very general in complex systems. Just imagine, for instance, earthquakes. In earthquakes, you are accumulating the stress. Yes, accumulating, and then it's a earthquake which makes the stress going down and going to zero. Lightning. Yes, in the lightning, you are accumulating electrical charges somewhere, and then it's lightning strikes. You reset everything to zero. So this grow and reset process is very, very general, and you find it very many times in nature. And this, you can write up then the master equation for this process, where you can go locally up, and then you introduce the probability also to reset. And what you can do, you can write up the Fokker-Planck equation for such a process. So you go in the continuum limit, you get again a partial differential equation, which was the Fokker-Planck equation. But the good news is that if you are looking for this process, for the equilibrium one or the stationary one, where the system tends after infinite time, the equation that you got is exactly solvable. So you always got a solution. So I can always tell you for a grow and reset process, what is the final equilibrium distribution? So what is the distribution where the system tends? And the beauty is then independently whether the grow rate and the reset rate are state dependent, I can give you an analytic solution. And let me just show you a video of what is this process. So you can go to any state, from any state, people so can go from, let's say, salary 0 to salary 1 to salary 2, salary 3, yes, but there is a possibility to reset. Yes, you just retire or die, go out from the system, and somebody else comes after you. So this is the grow and reset process. I find this video on YouTube, and it's showing very nicely. This is a performance which is made by a French artist, Jean Bourbeau, and you see he's illustrating my process. So you can go up or down, so there is this local grow process which you can go up or down, but sometimes you reset, yes? Your reset usually is not only 0, you can reset to another state, you can reset to another state which is not 0. So in fact, this is the model that I want to introduce and show you that this model is really applicable. So you have a lot of possibilities to apply it and to get the limiting distribution, the stationary distribution which is led this process which is leading to. Okay, so just let me give you some examples here why I consider that this model has a big applicability. So I told you that the equilibrium solution of this one, when you put here this term to 0, it's analytically solvable. So if you know the reset rates and you know the grow rates, which can be state dependent, you will get an analytical solution for the stationary distribution. And here I gave you some examples. So you see here are the possibilities that you can choose. Some possibilities you can choose for the reset rate, some possibilities you can choose for the grow rate, and what are the final distributions you arrive to. And the simplest process where the reset rate and the grow rate are state independent, so independently in which state you are, you apply the same probabilities, is leading you to the very famous exponential distribution. Yes, this is also a distribution which you encounter very many times in complex systems. When you say that the reset rate, for instance, is constant, but the grow rate is preferential, so it means the rich gets richer quicker, yes, the rich are growing quicker than the poor ones, then you go to the very famous Pareto distribution. And so on, you can get all the types of distributions, all the characteristic distributions that you encounter in complex systems. Now the question is always, if you want to model a complex system, whether your reset and grow model or the reset and local grow model can be applied or cannot be applied. I give you some applications that we consider here. I will not have time to go over it. Maybe I will go over the first one because that is interesting for any scientist, where you can see that science is not really much different from the whole society, because your citation statistics, everybody is interested how it's cited. And if you just look, your citation statistic is very similar with the Facebook shares. Yes, it's exactly so. Science, Facebook, they are the same, which is not too good for science. Okay. Then So you can explain all these distributions that you have in science for the citation distributions just by applying simply this grow and reset model. Then you can also understand wealth distribution in societies if you are applying this grow and reset model. Income distributions in the societies. You can understand deep distributions in networks. So in the last few years with Tomasz and some students of us, we are applying this model to many complex systems, and surprisingly find out that it's quite successful because we can describe all these distributions that we have seen. Let me speak about the first one a little bit. Maybe I have some few minutes to do that. But before that, I want not to miss this point. So my point for talking about this model is exactly for looking possibilities in biological systems. So if you are coming up for something that you think you have a good statistics and you want me to model it, I would be open to do it. I would be really happy to try it. And this is maybe the reason that it's so nice to be in an interdisciplinary atmosphere, because I can get some ideas, and maybe you get also some model for your experiments, and that would be very benefit for both of us. Okay, so let me just tell you a little bit about this first problem. I will definitely not have time to go on the other one. Just tell you some interesting things about your citations, and why your citations are very similar, and the citations of everybody, not only yours, is very similar with your Facebook shares. So it's exactly almost the same. So what we did here, first of all, this was a data mining, and we took up a lot of Facebook pages. Ronaldo, you have here the Facebook page of Ronaldo, the New York Times Facebook, NASA Facebook, Science also. And we look up also for citations which are from an institute, all the citations which are from authors from an institute, all the citations in a journal, let's say Lancet, and all the citations which a famous physicist, Stanley, is one of the fathers of complex system and physics. He has an incredible number of citations and an incredible Hertz index. And what we did, we plotted the probability to have a given number of citations. So what you have here, it's a probability density, in fact, so it's bigger than 1. It's a probability density to have a given number of citations over the average citations. Yes? So I take, for instance, Professor Stanley, I'm counting how many citations has with how many papers it has with 100 citations, how many papers has with 101 citation, and so on. And I am dividing for everybody this number with its average number of citations per paper. Yes? And here is the probability that this number gives something for me, the probability density. And you see that the curve that you are getting, first of all, for all universities, all journals, all authors, they are collapsing on a master curve. Yes? So first of all, universities are not really different from journals, and journals are not very different from scientists. And then you can do the same for Facebook. Yes? And you say that share in Facebook is somehow equivalent with citation, because yes, it means that you think that the information is important, you want to give it to others. And this is what you are doing with citation. You think that the paper you read is important, you put it in your reference list, so you want others to read it. And you see that everything what you get is uniform, and Facebook and science is totally the same. So you can fit both of them with the so-called Pareto distribution. Yes, so remember that one of the distributions I could get with the grow and reset model was the Pareto distribution. So you see that this Pareto distribution is in fact a power law distribution. This is a double logarithmic scale. So this is logarithmic, this is also logarithmic, and it means this is a power law. And the power law is valid on a huge interval. So you see that it's going on at least 4 orders of magnitudes, and here even more orders of magnitude. And now the question is why I think that the grow and reset model is good for this kind of citations. First of all, citations for a given paper does not go down. Yes, it's just going in one direction. You cannot lose citation. Yes, once you get it, you will have it there. And the same for the shares. If you have a Facebook post, the shares just can go on. You cannot use the shares anymore. So it's a growing process. Yes, you have the growth. But what is the reset? You can ask me. The reset is not so simple because when you die, your citations are not going away. It's still there. But what you can show that a dilution in the system, so if you are diluting the number of people that are in your statistics, is equivalent with a reset. So what happens in the scientific community and what happens for the users of Facebook, the users are increasing and the scientists are increasing and the number of papers that receive citations are increasing. And this dilution in the system, mathematically we can show that it's equivalent totally in our equation with a reset. So if you apply that there is a constant reset rate, the constant reset rate is valid if the increase is exponential, and this is the number of papers unfortunately are increasing exponentially. It's not a good news because they are not really selective. So we publish a lot and exponential growth is very, very, very dangerous. And that exponential dilution is equivalent with a constant reset. And what we are doing, we are also in the Facebook and also in science, we are citing preferentially. If something has a big number of citations, we will cite it. If a Facebook post has a lot of shares, we share it. Yes, usually there is a preferentiality. It's called the rich gets richer effect, or it's called the Matthew effect from the Bible. Those who got, they will get. Yes. So you see that this simple grow and reset model can beautifully, so the curve what I'm giving here is the result of the grow and reset model, yes? So the distribution that you get in experiments or you get it from the reality for Facebook and science, you can very nicely describe by applying this grow and reset model. And so on for other systems we have studied, wealth distribution in a society, income distribution in a society, distribution of degrees in a network, and so on. Thank you.
Thank you.
Thank you. This is very interesting. But I'm not sure I understood this graph. If you
Okay.
mind,
Can I repeat it?
yes. What do we have on the axis?
So again, so here, let's say that this axis is, I'm working for citations, okay? I speak now not for. So here are the number of citations for everybody divided by its average number of citations. So I take Professor Stanley. I'm calculating how many papers Professor Stanley has that has n citations. And I'm looking for all his papers, and from there I get what is his average number of citations per paper. Yes?
Okay.
And then I divide the n by this average number. Okay? And I'm representing the probability that Professor Eugene Stanley will have this number for a given paper. And here is the probability. So I'm counting how many papers he has with, let's say, 100 over this average. And I'm putting there the probability, which means I'm dividing with his total number of papers.
Okay, so the probability to have.
And just take it simple. The
And by an
probability.
average.
Yeah, exactly. And by its n average. That's right.
So I remember a few years ago, Barabasi was.
He has even more. Yes.
And he wrote this paper, The Science of Science, which was published in Science.
Yes, I know.
How is this fitting with what he published?
Yes, he's not doing this. What he's doing there, he wants to predict how many citations you have. So he was looking for a way to predict that somebody will have a given number of citations. So this was before them. So this was, I think, before the science of science. So he knew about that, but he uses that this is a Pareto type distribution. So in his prediction, he's using this one.
Oh, there we go.
Okay.
I mean, these can be used for prediction, right? Why not?
It's very useful, but it's you see, it's not a good message for science. It means that our selection process for what we cite is like Facebook, it's shared. And when you do share, you don't think too much. So, do you think enough when you cite somebody? This is the question. Or you cite somebody because it's cited.
And not because of the content, right?
Yes.
That's very important.
It's a positive feedback. I mean,
But also in Facebook share, wait, you have you have a filtering mechanism. You don't share everything. Yeah? You don't share bullshit usually. You just share, maybe you think it's interesting, you share. This is what science does. I think a paper is interesting, I'm citing it, but sometimes I'm citing just because somebody cited it. But this is with Facebook share.
But it's also lower probability you will find something which may be interesting but was not. It needs initiation point to be seen in the forest of that.
Yeah, but then once it's initiated, you see the growth becomes very quick because there is this preferentiality. So in the growth, there is this preferentiality.
Okay. Can I make this analogy? Since we work with lasers, it's the same thing with the emission of a laser. You start with spontaneous emission, and then the winner, the more in the cavity takes, so just because of the positive feedback.
Exactly. It's a positive feedback in the system. Exactly.
Yeah, could it be also a matter of the network actually? Because I mean,
you're right. This is also going on a network. So you see that the model that I'm implying here, it has nothing to do with what are the topology of the interaction. So this kind of master equations, they are a mean field model. So it means everybody can have information on the other one, or everybody can go from any state. So it's a mean field type approach. And once you go over the mean field type approach and you want to put it on a topology, these kind of processes, maybe you gain something, but you see, you don't gain too much. So this is what I say, definitely you will not be able to solve it analytically. So you use the beauty of the mathematics that here is analytically solvable. You could gain something, but the distribution is already known by such a simple approach.
I have one last question, please. So adding the reset term, what happens if I take the mu to 0? Shouldn't I recover the original equation?
Sorry?
If I take mu to 0 in the reset term, don't I?
No, the gamma is the reset term, sorry.
Sorry,
mu is the no, you don't get it back because in the original equation that you had the two-way diffusion. Now I have a one-way diffusion, I mean, just going up, okay? And if I would put back definitely the going down process, then I go back to the original. But this will not lead me to the original because this is a one-way growth and reset. Of course, I can make it two-way growth and reset, so I can go up and go down if you want, and reset, but then it's analytically not solvable. That's the problem.
Sorry?
Sorry, it was there.
Ready?
No, just can you do reverse actually?
Yes, you can. You
It's
can
the
just
same
go down
mathematically.
and jump up.
The same at
You can jump. The reverse going down and jump up.
Okay, so it's the same.
It's the same.
Yeah, first, this is for Eugene to mention that I was awake all the time for this presentation. Okay, and I would like to come back from social sciences a little bit to material science, and I would like to have a problem for this kind of mathematics. A normal distribution of magnetic nanoparticles, log normal, more exactly, okay. That means that the magnetic moments are not the same, yes? Now, they arrange them, we come with an ordering field, it's a magnetic field, and they arrange them in a linear chain, is the simplest case. And now the question is, where are going the small magnetic moments in this chain? And I will motivate why. When you apply a shear in the chain,
You reset them?
yes,
You reset
and
them.
influence highly the magnetic viscosity of this suspension. So it is very important where this chain will interrupted by the shear. That's why it's good for such kind of modeling.
Definitely.
Yeah, that was actually the
You
question.
apply reset process
Because
magnetic
really I
field.
don't know.
This is what you are doing. You apply reset process.
But it's very inspiring all what you presented. Thank you. Anyway, thank you.
Just one comment, because you always excite my imagination, you know. And I was also fascinated by the actor who was jumping from time to time. And the question is, what if this grow and reset model, instead of being just one-dimensional, that means to be just one guy jumping, to be two or three, each of them with different probabilities. But whenever anyone is down, everybody should be also done.
So definitely you can make in different variables. So you can take that somebody, let's imagine like that. You have not only income, you have wealth.
Yes.
So you have two coupled variables. And for
Yes.
both of them you can apply it. And one is interacting with the other one, but definitely if you
No, it
want to
should
do
not be, it should be independent.
and it's simple because
Yeah.
depends, everybody is decoupled from the other one,
Yes.
you can solve it, no problem.
Yes.
But yeah, but it's the same.
Yeah, yeah,
Just if
yeah.
you solve it solely, it's the
Yes.
same.
Uh,
But if you couple them, then it becomes interesting, but probably use the analytic solution.
no, I still would like to have another solution.
I don't think you will. So you,
No, without coupling.
you,
So
you
it
know
will
there is no free dinner. If you make the model complex, you lose analytic solutions. And that's the general way. You can just do it computation.
Thank you very much. Thank you. We have a coffee break, but I would like you to be back precisely a 3:45 because we'll have a Zoom presentation from Italy. So I think we should be 10 minutes back.
based on carbonic anhydrase inhibition, target identification, and validation. Please, Professor Supuran.
Yeah, thank you very much for introducing me. You have to habilitate me to condivide the screen because I'm not habilitated. I cannot show you my presentation.
Oh, yeah.
I think you have to put the red on me, on my name, to be able to.
Professor Supuran was the most cited author in the field of pharmacology and toxicology in 2013. It's okay?
Okay, now it will work, I hope.
Please.
Okay. So I wish to thank you for inviting me in this nice meeting, Eugen. I'm also very sorry that I was unable to come personally, but I had a quite busy period in this way, as I already mentioned. So I will speak about antibacterials based on carbonic anhydrase inhibition and the target identification and validation. I have to see how I can go. So the carbonic anhydrases are enzymes on which I work for many years now. They are catalysts of a simple reaction which is reproduced here. So the carbon dioxide is hydrated to bicarbonate and protons. Why do we need a catalyst for such a simple chemical process? Well, the answer is rather easy. This reaction occurs indeed without the catalyst, but it's very slow at the physiological pH. And carbon dioxide is generated in huge amounts in most organisms. You know that it's a gas, it can damage cells, structures of the cells, and all these create problems. This reaction also transforms two neutral molecules in a weak base and a strong acid, and as a consequence, it's one of the main used buffers in biological systems for regulate pH, and pH is quite an important parameter to be tightly regulated in most organisms. Maybe this explains why some of the carbonic anhydrases, either from vertebrates like humans or some bacteria like this extremophilic bacteria with an unpronounceable name, sulfurhydrogenic benzolense, are among the most effective catalysts known in nature. They are in fact second only to superoxide dismutase, which is the most effective enzyme known today. You see that the K-Km ratios are huge for all these enzymes. And this is also probably the reason why we have many genetic families including carbonic anhydrases in organisms all over the phylogenetic tree. Nowadays, we know 8 different genetic classes of carbonic anhydrases. They are nominated with Greek letters from alpha to iota. And you see that they are present both in prokaryotes and eukaryotes. Obviously, the enzymes in vertebrates and humans are the most investigated. They belong to the alpha class. I will not speak much about them today because I will try to show that in bacteria and other infective agents we can have the opportunity to intervene. In my laboratory we work with all these type of enzymes, and probably over the years we discovered and characterized around 150 new carbonic anhydrases from different organisms, mainly marine ones and bacteria ultimately. The E-type carbonic anhydrases, which were discovered in Plasmodium species, were also reported for the first time in my group. And we know quite a lot about the structural biology of these enzymes. Here I have a slide showing 4 different structures of 4 different carbonic anhydrases. An alpha class 1 from the humans, human CA2, a beta class 1 from pathogenic fungus, Cryptococcus neoformans, CAN2, a gamma carbonic anhydrase from an archaeon, it's called CAMP, and it's from Metanosarcina thermophila, and the zeta carbonic anhydrase from marine diatom. with the name Thalassiosira slowly. You may see that the overall organization of these enzymes is quite different. So here for the alpha class, we have a globular protein with a rather wide and open active site, which has a hydrophobic half and a hydrophilic one. So this is a unique example among enzymes in which we have this bipolar organization of the active site. Most carbonic analyses are metalloenzymes, and the metal ion is crucial for catalysis and is found at the bottom of the active site cavity, shown here in yellow. The beta-carbonic anhydrases, which you will see later, are quite widespread in bacteria, are dimers. The active site is at the interface between two monomers, and it's again shown here as a rather long channel, so it's not so wide as the alpha-carbonic anhydrases. But again, the metal ion, which is zinc or other metal ions can be present, is at the bottom of the active site, and we have bipolar structural organization. This is more or less the same in gamma carbonic analysis, which on the other hand are trimers. So we have 3 active sites at the interface between two monomers, shown here again, and the active site is shallower compared to the other ones in gamma class enzymes. Finally, the zeta carbonic analysis of cadmium-containing enzymes. This is quite unexpected because cadmium is usually associated with high toxicity in living systems. Here instead it has catalytic role, it's not toxic, so it performs exactly the same thing that the zinc and other metal ions perform in other carbonic anhydrases, which I show here. In fact, in most of these enzymes, there is a zinc hydroxide or a metal hydroxide species, which is the nucleophilic, which attacks the carbon dioxide bound in a hydrophobic pocket not far away, with formation of the bicarbonate, which is in this 3-1 biparallel coordination of the zinc. I didn't mention, but you see here that the zinc is coordinated by 3 amino acid residues like 3 histidines or histidine and cysteines, depending on the enzyme class. Finally, this bicarbonate adduct is not very stable, so it can be displaced by an incoming water molecule with bicarbonate liberated into solution, and the formation of this form of the enzyme is called the acidic form of the enzyme, with water as the fourth ligand. Finally, in order to generate the nucleophilic species, a proton transfer reaction must occur, which is in fact assisted by amino acid residues placed in the middle of the active site, like histidine 64 in alpha-carbonic anhydrases or other residues in other classes. And all this cycle occurs more than 1 million times in different isomers. So it's really a hugely effective catalyst. Now, returning to the bacteria. So carbonic anhydrases have been discovered in bacteria already in the 60s and 70s, but they were not characterized till recently. Nowadays, we know, or better to say, till 2020, we knew that there are 3 genetic classes present in carbonic analysis: alpha, beta, and gamma ones. And in 2020, we discovered, and other colleagues discovered a 4th class, which is called iota carbonic anhydrases. I already mentioned something about the alpha, beta, and gamma in the previous slides, but anyhow, they are all metalloenzymes, whereas you will see at the end of my talk that iota carbonic anhydrases seem to be non-metalloenzymes at all. The coordination of the metal ion, which can be zinc, iron, but it's also cadmium in some cases, it's quite diverse, but anyhow, the catalytic mechanism that I showed previously is valid for all of them. And you know much better than me that the bacteria can be divided into gram-positive and gram-negative, and there are obviously different differences between them, but from our point of view, it should be mentioned that the gram-negative have 2 membranes, an outer membrane and an inner plasma membrane, and this periplasmic space, which isolates the cytoplasm from the rest of from the medium. Whereas in the gram-positive, we don't have these 2 membranes, we just have the cytoplasm, the plasma membrane, and obviously the peptidoglycan cell wall. And you know very well that most antibiotics, in fact, target the peptidoglycan biosynthesis, and there is a huge problem of resistance to currently used antibiotics. So there is a need to try to find different types of compounds which inhibit the growth or eventually kill the bacteria. So we started to look at bacterial carbonic analysis more than 10 years ago, and we observed something quite interesting. So if we look at the genetic classes of carbonic analysis in the gram-negative and gram-positive bacteria, we see quite a difference. So there are some bacteria, some gram-negative bacteria which contain only alpha, others contain alpha, beta, and gamma, and very few contain only beta and gamma. Whereas for the gram-positive, basically there is no alpha-carbonic anhydrase in any bacteria, and there are only beta and gamma or only gamma. So we try to understand why this occurs. I should also mention that some of the very few organisms that were completely devoid of carbonic anhydrase are two bacteria belonging to the Buchnera and Rickettsia species or genus. And probably they don't contain carbonic anhydrase because they are symbionts with other organisms which can provide them bicarbonate or pH regulation in the niches in which they live. So our initial questions were these. Why are alpha-carbonic anhydrases present only in gram-negative and not in gram-positive bacteria? What is the physiological role of these enzymes in the bacteria? Why are alpha-carbonic anhydrases not present in all gram-negative bacteria? And what is the role of the recently discovered iota-carbonic anhydrase class in bacteria, which is an argument that I will treat at the end of this talk, as I mentioned. So what we did together with Clemente Capas, our colleague that Michaela and Eugen know well because he participated in some of our common projects, we did a phylogenetic analysis making the alignment of human carbonic anhydrases belonging, as I said, to the alpha class and some alpha class bacterial enzymes, such as, for example, Helicobacter pylori, carbonic anhydrase Librium cholerae, sulfurylhydrogenidium, and so on and so forth. And basically, if you look at the amino acids which are important for catalysis, like for example the zinc ligands shown here in blue, shown here in red, histidine 94, 96, 119, or the proton shuttling residue shown in blue, or some other acids like for example 106 and 199, glutamate and threonine, they are basically conserved in all these enzymes belonging to humans and bacteria, which basically had a common ancestor probably billions of years ago. However, if you look at the N-terminal sequence, we saw that a signal sequence is present only in the bacterial alpha-carbonic anhydrases and in the secreted and mitochondrial human carbonic anhydrases. And we know that all these signal peptides basically orientate these proteins towards a certain place of the cell. In fact, this N-terminal signal sequence is just the way of the protein to be localized in the periplasmic space that, as I mentioned, is present only in the gram-negative bacteria. So now we have already a partial reply. Why are alpha-carbonic anhydrases not present in the gram-positive? So in gram-positive bacteria, we don't have the periplasmic space, so that's the reason why we don't have alpha-carbonic anhydrases there. And then we look at those alpha-carbonic anhydrases which are relevant for bacteria involved in infection. For example, in Helicobacter pylori, it's known that the periplasmic alpha-carbonic anhydrase is participating to the acid acclimatization of the pathogen. I will speak about this in more detail later. In Rhasconia eutropha, it assists the transport of bicarbonate across the cell membrane when this bacterium grows in autotrophic conditions. Finally, in Vibrio cholerae, the bicarbonate it's a factor which increases the pathogenicity, and in fact, the periplasmic carbonic anhydrases increase the concentration of bicarbonate outside the bacterial cells in order to induce virulence. So the role of periplasmic alpha-carbonic anhydrase is mainly in the adaptation to habitats, and this probably explains why we don't have it in all microorganisms, because not all of them need to live in highly acidic conditions as Helicobacter pylori, or in more alkaline conditions like Vibrio, and so on and so forth. So most of the carbonic anhydrases, as I show later, are in fact involved in the survival of the microorganism, so in metabolism. And indeed, the beta or gamma carbonic anhydrases, which are present in almost all bacteria, are in fact giving one carbon unit in the form of carbon dioxide or bicarbonate for metabolic processes such as carboxylating processes and so on and so forth. In gram-negative, where we have the periplasmic space, in some cases when the bacterium needs to be acclimatized in different niches, they can contribute to this buffering effect either outside or inside the cell. In fact, we know from knockout experiments that, for example, if you do the knockout of the beta or gamma carbonic analysis in Escherichia coli, we will have a reduction or even an inhibition of the bacterial growth, but only at low carbon dioxide concentration. Why this occurs? Because obviously at high concentration of carbon dioxide, the non-catalyzed reaction can in fact supply bicarbonate or H+ ions for the bacteria to go. Another example is Pseudomonas aeruginosa. Also here, we have 3 carbonic anhydrases in this bacteria. If we do the knockout of the beta carbonic anhydrase called PSCA1, we have a reduction of the soft tissue calcification, which is one of the processes by which Pseudomonas induces disease in humans. So the idea that I had already in 2011 in this review article was, if we inhibit carbonic anhydrases in some of the pathogenic bacteria, we will of course have an effect on the carbon dioxide and bicarbonate concentration, and this may lead to the bacterial growth impairment of the growth or growth defects. So can we generate new antibiotics using this approach? And you will see that in fact the answer is yes. We worked hard for the last 10 years with different colleagues in Italy and USA, and I think we were able to show that this is in fact possible. So initially there was some data in some pathogenic bacteria such as Helicobacter pylori, where it was shown that the carbonic analysis is essential for acid acclimation. As I mentioned, in Vibrio cholerae, it's involved in the production of the cholera toxin. In Mycobacterium tuberculosis, it's a virulence factor. In Pseudomonas aeruginosa, it's involved in soft tissue calcification, whereas in Brucella rujulensis, it's cellular growth. But of course, one important issue is that the carbonic analysis are also present in humans. So if you want to have drugs which don't have bad side effects, we have to take into consideration that in humans, there are many physiological and also pathological processes in which these enzymes are involved. And here's just an enumeration of them, and you see that many of the human carbonic anhydrases are in fact therapeutic targets for edema, so diuretics, anti-glaucoma drugs, epilepsy, hypoxic tumor, obesity, and so on and so forth. So one of the main issues to address this fact that the carbonic anhydrase inhibitors can be indeed anti-infective drugs is to try not to alter very much the drug targets from the human organism. This is something that I showed several times, but I will show it again here. So we have this is an alpha carbonic anhydrase, a human carbonic anhydrase. We have 4 general inhibition mechanisms, which are shown here with crystallographic data. So this is the surface of the protein, the active site is the hydrophobic hydrophilic half, the proton shuttling residues, and you see that we have inhibitors which coordinate to the metal ion like this sulfonamide called the cytosine. It binds nucleus in the active site. We have inhibitors which anchors to the zinc-permeated water. So this is phenol, hydrogen bond is formed between the OH of the phenol and the zinc-permeated water. Of course, the scaffold of the inhibitor may participate in interactions and usually participates in interaction with the hydrophobic hydrophilic alpha enzyme. We have the coumarins, which have a very nice inhibition mechanism. They are hydrolyzed at the lactonulin with formation of hydroxycyanamic acids, and they bind at the ends of the active site cavity. I repeat, this is all crystalography. It's not any docking that I showed here today in my talk. Finally, we have an out of the active site inhibition mechanism, exemplified here by this benzoyl, by this benzoic acid with benzyl sulfoxide moiety. So this compound binds in this adjacent pocket at the entrance of the cavity, and through the carboxylate moiety blocks the proton shuttling residue, which is shown here in green and here in detail. So through a bleaching water molecule, it forms a strong hydrogen bond, and the ECD64 is blocked in just in one orientation. It is unable to participate in the proton shuttling cycle, and basically the entire catalytic turnover is blocked. And over the years, so starting with the 90s when I started to work in this field, basically we knew just sulfonamides, phenol, and some sulfonamide variations as inhibitors of carbonic anhydrases. Fortunately, from that period, many other different chemotypes have been discovered, like coumarins, dithiocarbonates, this out of the active site inhibition that I just mentioned, phosphoramidates, linkidrins, xanthates, polyamides, cellular, and so on and so forth. And I'm rather glad to tell you that from this point on, all these new chemotypes that have been discovered were discovered in my laboratory in Florence. How I started to work in this field and why I'm interested in bacterial carbonic anhydrase inhibition. Well, probably you know this scientist, Jan Puskás was my mentor for 1 or 2 years when I was in Romania at Transdisciplinary, and probably some of you know that he was using to treat peptic ulcers with acetazolamide, which was something that not everybody thought it's valid. However, it was indeed valid, and ironically, the fact that he was right was demonstrated just by his biggest scientific enemy, a medical doctor from UCLA. So the idea of Puskás was that acetazolamide was inhibiting the carbonic anhydrase in the stomach. So there are a lot of isoforms, but mainly CA1 and 2 in the stomach. And the inhibition of these isoforms obviously can lead in principle to a reduction of the hydrochloric acid. This colleague from UCLA in USA showed in the late 90s that in fact acetazolamide is inhibited Helicobacter pylori, which, as you know very well, is the pathogen which provokes gastric cancer. So basically, Puskás used a rationale which was in principle not valid because he did not inhibit human carbonic anhydrase, but in fact the inhibition of the Helicobacter pylori enzyme leads to the killing of this pathogen and in fact to the healing of the ulcers. So why I did all this long introduction? Because in fact it is now known and already mentioned that in the periplasmic space of Helicobacter pylori, which is a gram-negative bacterium, we have alpha carbonic anhydrases. These alpha carbonic anhydrases are involved in the buffering and in the adaptation of the bacterium to live in this highly acidic pH within the stomach. And it's not only the alpha carbonic anhydrases, but also the urease, which is generated from the activity in the cytosol. So urea is decomposed in carbon dioxide and ammonia. Ammonia can be transported and can participate to the buffering, but also some of the CO2 and bicarbonate can be used to biosynthetic processes within the cytosol within the cytoplasm of the bacterium. So this is the mechanism by which carbonic anhydrases participate to the activation of Helicobacter pylori in the stomach. If we intervene in one of these processes, for example, blocking the urease or blocking alpha or beta carbonic anhydrase, the entire mechanism of activation is not occurring, and basically the bacteria cannot survive in that harsh environment. And together with a group in Australia, I was able to crystallize the alpha carbonic anhydrase from this bacterium. It's a dimer. I repeat, it's an alpha carbonic anhydrase, so zinc and glycine residues. Here we have even acetazolamide bound to this enzyme. You may see that the overall shape of the molecule is not very different from that of other bacterial carbonic anhydrases, such as, for example, the one from Neisseria, which was reported in the late 90s. That from our bacterium from Yellowstone, so the one with difficult name, Sulfur-Tiprogenidium, or another, this is Thermus ammonificans, a bacterium which was crystallized by other colleagues. And we have several inhibitors which were crystallized to this enzyme. So we understand, for example, why acetazolamide is quite an effective inhibitor, whereas metazolamide was not. So it's just a clash between a methyl group present, so this methyl group present in metazolamide, which is not present also in acetazolamide, which leads to a 10-time difference of inhibition between the two. We have also some data for the inhibition of other compounds of Helicobacter pylori. Probably the most interesting example is compound, I think it's compound 3, because it's the only one which is more effective for inhibiting Helicobacter pylori enzyme over the human enzyme. Yeah, 8 versus 34. And again, by crystallography, we understood why this happens. So basically, in the human enzyme, we have this alpha helix, this loop, which is not present in the bacterial enzyme. In the bacterial enzyme, it's just a small loop. And the tail of the inhibitor makes a clash with this loop, where it's not interacting in such a negative way with the other one. I'm afraid I can if you want to interrupt me, just let me know. But basically, I think I will get it wrong. So we have other examples of such bacterial carbon analysis that we have characterized. This is the Vibrio cholerae alpha carbon anhydrase, the beta one from the same pathogen. This is of course a different structural organization because, as I mentioned, this is a tetrameric. It's a dimer of dimers enzyme, and here you see the active site with one histidine and two cysteines coordinating the neuroline. And in fact, in this review, we propose in more, let's say, with more courage the fact that the carbon analysis inhibitors may be antibacterials, because there were a lot of recent data in the late 2020 and in the last 2 years. And probably one of the most important work is this from our friends in USA, Daniel Flaherty and Mohamed Salim, because they optimized compounds based on acetazolamide for the inhibition of microbial-resistant enterococci. And in fact, they show that acetazolamide and some of its derivatives have indeed a very strong in vivo antibacterial activity. And together with us, we started to work on Neisseria gonorrhea alpha-carbonic anhydrase, because this is an enzyme which has been crystallized quite some years ago, and we have also some inhibitors which are bound to it, so they can be used for the rational enzyme. Here's the adviside for the human and for the bacterial enzyme, and you see again that the human enzyme has an extra alpha helix, which is not present here, which can be used for doing better inhibitors. So together with Flaherty and Sabine, we were able to design some of these compounds. So this is a fluorescent probe that we used for titrating the enzyme. And then, based on the acetazolamide scaffold, we have different compounds with different substitution, which show some inhibition of growth in the low under 1 milligram per milliliter amount. So which is quite good because, in fact, there are few compounds which are like this. Not only, but we are able to measure MICs also under normal CO2 and high CO2 conditions, and you see that most of such compounds still maintain a good efficacy in these different conditions. So we have the structural relationship of this type of compounds. I will not enter into detail because I will try to arrive at the end, but what's very nice is this is just March 2022. So Mohamed was able to show that in mice infected with Neisseria gonorrhea, which is drug resistant to most antibiotics, the treatment with acetazolamide as rather low concentration is indeed very much anti-infective. So basically it's a proof of concept in vivo in an animal infected with this pathogen, not only in what we showed in cell culture, in which we obtained by nice data. So basically, this is a quite important validation that I recently. If I have 2 or 3 minutes, I would speak about the IOTA, otherwise I can also stop here. I don't want to be longer than my time, so please let me know when I can continue or not.
You can.
Yes, okay.
You can continue.
Okay. So the IOTA carbonic analysis were reported in 2019 by a group in Marseille, Gigi Gontero, and it was quite interesting because they reported to be manganese enzymes, so they are found in diatoms. Then, together with Clemente, we also found that we have IOTA carbonic analysis in many bacteria, so we characterized one in Burkholderia territory. And we obtained the fact that it's not manganese, but it's zinc enzyme. So we don't know, at that point we didn't know whether Gontero or us we are right or not. To make things even more interesting and nice, in fact, a Japanese group characterized a new type of carbonic anhydrase that we initially called kappa, the kappa class, but in fact it was shown to be a IOTA carbonic anhydrase. But this IOTA carbonic anhydrase had no metal, and they did the executive structure. So this is the first structure. Basically, there is not any metal ion in it. So at that point, obviously, we started to be quite concerned whether this is an enzyme without metal ion, with manganese, or with zinc. So we made this review article last year in October or November, and again we used phylogenetic analysis. So basically, the enzymes which are characterized are 2 of them, and they are one from an alga, the other one from a bacterium. And in many bacteria, we have IOTA carbonic anhydrases which cluster quite differently from the ones with no metal ion. But then we tried to understand whether this crystal structure that was reported by the Japanese group has some similarity in other proteins. And in fact, there are proteins called NFT2, which are quite widespread in many, many different types of tissues and with different types of activity, which all of them have this type of butterfly-like structure. The one of them, which is the crystallized iota carbon anhydrase, is this B, and excuse me, C, and the organism from which it has been isolated is a cyanobacterium, which is called anabaena, whereas the microalgae is called Biodynamics natans, and it's shown here in. So anyhow, you see there are quite a lot of similarities between these structures. But as I mentioned, there is no metal ion bound to them. However, the anions like bicarbonate, iodide, or gas carbon dioxide, so bicarbonate or even small molecules, do bind in an active site, which is in fact quite similar to the active site that I showed for you for other carbon anhydrases. But in fact, this enzyme does not use metal nucleophile, so metal hydroxide nucleophile for the catalysis, but it's using a water molecule which is anchored to a catalytic triad formed from a threonine, a serine, and a histidine. So this is the catalytic triad in iota carbon anhydrase. This is bicarbonate, which is bound, which is seen in the crystallographic structure. Here's iodide, which is bound. Here I don't remember what it is, maybe it's carbon dioxide. So basically, it's a very nice example of an enzyme which exerts exactly the same catalytic function as the normal carbon anhydrase, but without a metal hydroxide. And together with Nainier and Mohammed, we wrote this review on bacterial carbon anhydrase quite recently, so this was published in 2021, in the hope to try to attract the attention of these underexploited drug targets for obtaining antibacterials. My conclusion is that the various carbon anhydrases belong to the diverse families are present in pathogenic bacteria, but also in fungal, protozoan, and nematode pathogens. Many such enzymes were cloned, purified, and now I should say quite validated, some of them. The inhibitors belonging to various chemical classes have been discovered, and some of them are also isoform selected for the pathogenic of human enzymes. We have now in vivo validation, at least for the Neisseria and maybe also for the condition resistant enterococci. There are still some problems with inhibitor selectivity, but anyhow, the bacterial series are, in my opinion, quite promising anti-infective targets. So I thank you very much. I will interrupt the conclusion and continue to
Thank you very much.
the questions I'm available.
Thank you very much, Professor Supura. Please, question, comments? I would like to ask something, please. 40 years before, or maybe years before, coumarin was used in order to treat pipe tobaccos. The concentration in these pipe tobaccos was enough to inhibit carbonic anhydrase or not?
No, so yeah, of course, some coumarins, in fact, they are B-coumarins derivatives used for blood coagulation problems. So they are anticoagulants, old generation anticoagulants, but their structures are quite diverse from the coumarins that show carbonic anhydrase inhibitory properties. In fact, when we reported the coumarins in 2009, we tested those compounds, and they don't inhibit carbonic anhydrase because they are too bulky to enter within the active site. As I mentioned, the coumarin has to be hydrolyzed at the lactone ring, and this hydrolysis occurs by the zinc hydroxide mechanism. The coumarins in clinical use for blood coagulation are in fact too bulky, and they cannot enter into the active site.
Thank you. And concerning the urea and the Helicobacter pylori, Helicobacter pylori has the capacity to hydrolyze urea in CO2 and ammonia. And this is used labeled urea with stable isotopes, for example, C14 or nitrogen 15, in order to diagnostic infection with Helicobacter pylori. And this test because humans don't have enzymes to metabolize urea, and this can be linked with the carbonic anhydrase properties of Helicobacter pylori or not?
So yeah, you are absolutely right. The urea is present in Helicobacter pylori. It's used for the test because the ammonia which is liberated can be detected, and it's a quite effective test to show the infection with Helicobacter pylori. Basically, the ammonia which is generated in very small amounts in the stomach of the patients which are infected, it's, as I showed, it's used together with bicarbonate to make a kind of buffer, a very localized buffer, to be able for the bacterium to survive in the stomach. Of course, as a side effect, some of this ammonia can be exhalated and can be detected. But the role, the physiological role that this urease and carbonic anhydrase possess is just that to make it possible for the bacterium to live at pH 1.5 or 2 in the stomach.
Thank you. Other questions? Thank you once
There is
again.
one question.
Huh?
There is one.
There is a, please.
I was interested about potential resistance to these compounds. For example, do you have some experiments related to the change in expression of these enzymes in the presence of the tested compounds, of the tested inhibitors?
I didn't hear very well your question. I apologize. Maybe you can speak closer to the microphone.
Unde e microfonul?
Okay. I was wondering about the resistance to these compounds. And I was asking if you have some experiments related to the changes in the expression of genes for these enzymes in the presence of the tested compounds.
So if I understood well your question, you asked whether there are compounds which can interfere with the gene expression of bacterial carbonic anhydrase.
Yes.
No, as far as I know, no. Even in the case of human carbonic anhydrase, so there are some reports, for example, in the there are some carbonic anhydrases which are present in tumors. They are overexpressed in hypoxic tumors. And there are some recent reports that some natural products, for example, like flavones or resveratrol, can interfere with the gene expression of these proteins in humans. As far as I know, there is nothing like that for the moment reported for bacteria or other pathogenic species.
No, but the bacteria could adapt, let's say, to these inhibitors by increasing the expression of these genes. So they can serve their metabolic needs and become resistant. What do you think about this?
The data that we have is that we inhibit the activity of the enzyme, so we don't interfere with the expression of the gene of the genes encoding for carbonic anhydrases. We just interfere with the production of the reaction products, so bicarbonate and H+. And this is detrimental for the growth of the pathogen. Either because it's not making it possible to survive in that niche, for example, it's too acidic or too alkaline, or because it's not having enough carbon sources for the metabolism. You know very well that the bacteria proliferate quite quickly, so they need a lot of nutrients. And in some cases, the nutrients are just carbon dioxide or bicarbonate, which can be used for the growth.
Yes, but at the same time, they could very quickly switch to a metabolically persistent state, so they are not multiplying so easily, and also they could shift their metabolism. And this is the challenge. Because, for example,
probably it can occur. So the data that we have in vivo for the moment are not valid. So we have data in vivo for Helicobacter in Australia and for Neisseria in USA, and the colleagues with whom we collaborate also did experiments to try to grow these bacteria, these pathogens, in the presence of high concentration of these, let's call, new generation antibiotics. Even in some cases they are not new because acetylcholine from the 50s. And they didn't see mutations in the gene of the carbonic analysis, which is a good sign. So this is also valid for Helicobacter pylori. So we have, I didn't show it today, but we have a very nice model in Melbourne, in Australia. So even after several months of being cultivated in the presence of etoxolamide, another carbonic anhydrase inhibitor, there were no mutation in the carbonic anhydrase gene, and this bacteria were still susceptible to be inhibited, their growth by the inhibitor, which of course does not occur with other antibiotics. So unfortunately, the resistance is developing very, very quickly if you grow them in suboptimal concentration of the antibiotic. You know that better than me. So yeah, there may be metabolic shifts, but in this case, we don't have any proof of that, at least with the inhibitors with which we work for the moment.
Thank you.
Thank you. Thank you very much.
Thank you very much. It was a pleasure to talk to you and see you from online, and I hope to be able to see each other also soon. You are kindly invited to visit us in Florence. Unfortunately, I will be unable to stay longer because I have another commitment, but I wish you all the best. Thank you very much.
Now I would like to invite a team from University of Medicine and Pharmacy Carol Davila Bucharest and Cantacuzino Institute. A team Andrei Alexandru Muntean, Madalina Maria Muntean, Nicnea Ioan Gabriel Popa, Bogdan Alexandru Miron, Vlad Tudor Popa, and Mircea Ioan Popa. Please.
Dear all, hello, my name is Andrei Alexandru Muntean. I am extremely honored to be here. So I'm extremely honored to be here, and I would like to thank everybody that has permitted this event to happen today and has invited us here. I feel very welcomed here, and I feel very close to home, both that we work at the university and the Cantacuzino Institute, which is not far away, but also much of the research that you will be seeing here has taken place right across the street in the Popescu Institute for Physical Chemistry. So a bit of my heart is always will always remain in that institute as well. My talk today will be about moving towards near real-time antibiotic susceptibility testing and antibiotic resistance mechanisms detection. And so the index of the presentation will be the problem of antibiotic resistance, which I'm sure is known to everybody. I will just show a couple of slides to point out why this research is important for Romania especially. Then I will be talking about screening and about confirmatory testing. And part of it will be conventional, so what is known and what is actually practiced today, or possible to be practiced in a microbiology laboratory, and some of it is novel, original research that we have done, as I said, as a partnership between the three institutes that I have mentioned. So the problem of antimicrobial resistance can be very clearly seen here. Of course, we talk about a lot of bacteria. Some of the worst, let's say, bacteria are those that are very much implicated in healthcare associated infections, and you see one of these bacteria, Klebsiella pneumoniae, which has very high resistance rates, and you can see a bit of a dynamic from west to east that there are countries in which antibiotic resistance is very much of a problem. This here, we show the resistance to third generation cephalosporins, which are the workhorse of the hospital environment antibiotics, but we can, of course, continue to carbapenems, which are some of our last resort antibiotics. Now, this has to be nuanced a little bit today because we do have novel drugs and there are, of course, others that will be coming along, but carbapenems are, for today, one of the mainstay and one of the last resort antibiotics that we have. So if you continue to another hospital-based pathogen, Pseudomonas aeruginosa, we can see that, again, we have very high resistance and Acinetobacter species. Again, we see in these reports from 2019, these are the latest that we have, we see that this pathogen is also a problem. You lose that west-east heterogeneity, you gain a little bit north to south in this case. So, gram-negative bacteria can become resistant to antibiotics in a lot of ways, and I will just be concentrating on a few of them, although as laboratory scientists, as doctors in the laboratory, we are also interested in picking up every type of resistance, not just beta-lactamases, but beta-lactamases pose a particular problem because they are most often plasmidic and they escape from one bacteria and they go and populate and just give off basically free recipes for antibiotic resistance to other bacteria. So once a bacteria with a beta-lactamase with a plasmid enters your institution, then it can spread and it can basically continue to quote-unquote infect, if you will, render resistant other bacteria that may be there. So what is there to do in front of something like this? Well, it's screening, and we will discuss if we're doing too little or too much in the sense of guidelines, confirmatory testing, expertise, and action. Nothing that I will be speaking about today has any relevance in the absence of action. So screening, conventional screening is done through conventional antimicrobial susceptibility testing, and this, as probably most of you know, is done through the Kirby-Bauer method through a agar plate with antibiotic discs, and this has been standardized. Or we can do it in a liquid broth medium, and we have screening guidelines and we have therapeutic guidelines, and they are not exactly one and the same. So there might be a little more leeway for screening in the sense that some bacteria might not be quite resistant, but they still might harbor some antibiotic resistant genes, and we want to head them off as quickly as possible. So are we doing too little? If we look at the evolution of the guidelines, which has evolved from 23 millimeters up to what is now 28 millimeters, we see that even though the goalpost has constantly been moving, we still lose a couple of bacteria which still harbor different antibiotic resistant genes that we cannot catch with today's guidelines. And of course, everything that we do in medicine and research is a balancing act between specificity and sensitivity. And here we can see what escapes even if you apply both of the guidelines. So there is a number of bacteria that will still always escape the screening mechanisms that we have today. What if we go too little? Well, we see that most of the we see that most of the ones that escape are OXA48 type carbapenemases and just very, very few others, especially a couple of metallobeta-lactamases. Why are we interested in this? Because the European Center for Disease Control and Prevention has done research and showed us that we are actually missing some of these resistant mechanisms, and they are very quietly spreading in hospitals and in other environments. So it's clear that as of today, we are not able in a clinical fashion, that is to say, in something that is done for public health, for the treatment of the patient in the hospital, not research purpose only, but actual guidelines and action in the hospitals and in the laboratories. We are missing some of these bacteria. because our methods are not there. So these are very, very particular organisms. What can pique our interest? Because we're talking about dynamics, and we're talking about growth of bacteria, and we're talking about the idea of getting these informations as fast as possible. Well, recently, and this is extremely interesting, this actually came out, the new version of this came out in April of this year. We can actually test for, and we can screen for carbophenamases starting from the blood culture of patients, and we can do this in around 6 hours time. And this has been published, and these are guidelines from the European Union Commission on Antimicrobial Susceptibility Testing. So this is something that actually can be done in hospitals right now. What was our approach? And this is research that we've been doing since about 2014 and onwards. So we've been working with the Institute for Physical Chemistry, and we've been using a device called an DSC, differential scanning calorimeter, which we've altered in a sense. We've reprogrammed it as an isothermal calorimeter. I know we're losing some of the functionality, but we have been culturing bacteria in this isothermal microcalorimeter, which allows us to have a reference ampule and a test ampule, and we will grow the bacteria inside. And growing bacteria in itself is extremely interesting because you have a lot of information about their metabolism, and you can differentiate between species, and you can get all sorts of insights on the basic microbial growth. But even more interesting is how you can apply antibiotics to the bacteria that you're growing inside the calorimeter and see their reaction. So reaction, in this case, I'm talking about real-time interaction between the bacteria and the antibiotic. And in fact, what we're seeing in the microcalorimeter is generation of heat. And so because we're seeing generation of heat, we're seeing metabolic activity, so we're seeing life. When heat production dies down, life stops. And of course, this is not a perfectly valid statement, since there can be dormancy states in which no great amount of heat is being produced. But in the case of very active, very fast-growing bacteria, like we would have Klebsiella pneumoniae over here, when the heat production dies down, then the bacteria is actually dying, and basically that means that the antibiotic is effective, the antibiotic is working. So here we have a number of Klebsiella pneumoniae species, and opposed to what has been previously done in literature, where you would cultivate a tiny amount of bacteria and wait for them to grow, we would challenge bacteria. And challenging bacteria means we would have a lot of bacteria from the beginning, and we would add antibiotic over it, so that we know their dynamics would be exponential, and we see what happens with it. And seeing what happens with it, here you see very long registrations of the growth curve, 30 hours long, but you can scale it down to what happens in 1 hour. In 1 hour, you see the effectiveness of the antibiotic, you see the reduction in what is normally to be the bacterium growing in the medium. So you know you have bacteria, you know it should grow. How is the antibiotic affecting it, and is it affecting it enough so that you consider the bacteria is actually dying and is actually inhibited? So this is manual processing of the thermograms, and you can see that we've used a carbapenem antibiotic with carbapenemase producing enterobacteria, some of them actually being resistant, and you can see the death of the bacteria, so to say, and we've normalized everything to go in a 0 point, which is completely arbitrary at this point, but we normalized everything to go to 0 and then see what happens. And as you can see, in most cases, so in the, let's say, most susceptible of the 3 Klebsiella pneumoniae that we have examined, the OXA48, at 160 micrograms per milliliter, we have the death of the bacteria, and this is quite evident within the hour. And as you can see here, there was no regrowth afterwards anyway. This is interesting, but it's even more interesting when you get a computer to do your job. So it's quite easy to do a small computer program that will allow you to stabilize and detect when actual isothermal calorimetric growth is being obtained, and then it can calculate for you a lot of parameters, even kinetic parameters, and you can see here lots and lots of these experiments which were used to actually generate the small computer program. It does quite a good job, so normalizing information for you, so this could be automated, and you can have computers just flagging positive vials for you, like it would happen in any medical laboratory that grows blood cultures. You would have an apparatus that will grow bacteria in the presence of antibiotics and will flag a positive or a negative. And this, very interestingly, can be done within an hour. In medicine, it is difficult to go and negate lots of what we know, so the MIC, all of these parameters that are used in clinical microbiology. This is really not something that we use here. It's basically, it's thought of as a screening mechanism, but as a screening mechanism, it works absolutely great. And here you can plot all of those interesting growth kinetic models and actually study the bacteria. On the other stance, would it be too much to consider that growth in the presence of antibiotics investigated through microperimetry would flag all too often? Would we get too many bacteria flagged for potential problems? Well, that depends on what you're looking for. If it's for the patient in itself, him or herself, you would not, because basically it is what you're going to ultimately use to treat them. If you're looking from a public health standpoint or from a hygiene department standpoint, you would be interested in not only what the bacteria has as a resistance level, but also why is it flagging positive and why is it producing resistance to the last line of defense. And so are we screening too much? Well, there are mechanisms in the laboratory that can compete with carbapenemases in leading to carbapenem resistance, and one of the most important would be the Ambler class C, the AMCs, the chromosomal or plasmidic cephalosporinases, which can be present naturally in some bacteria and acquired in others. And if we see the bacteria which actually do screen positive for a carbapenemase through conventional methods, we see that almost a third of them, or maybe a fourth of them, are indeed negative. So are we screening too much? Well, it depends on how you're looking, and this is how this test would be done in the laboratory. You would have plates with different types of antibiotics, and you would flag different types of antibiotics, which we would use as markers, and you would get the idea there might be something there. So the cefoxitin, this here is very telling of this particular isolate. And then we would use different, let's say, less than conventional media. In this case, one that has cloxacillin in it that will actually inhibit the AMC mechanism, and you can see how the diameter inhibition springs up around cefoxitin. All of these three plates are from the same organism. These are all tested on Muller-Hinton's media. This is cloxacillin media. Here again, we have a combination of at least two mechanisms. One would be the AMC, which this particular Enterobacter cloacae is overexpressing, and also an ESBL. This leads to resistance to most antibiotics, and it flags as suspicious for a carbapenemase since the diameter around ertapenem, the most sensitive of the carbapenemases, of the carbapenems, is reduced. But once we plate it on a Muller-Hinton cloxacillin plate, we see that the zone of inhibition around ertapenem grows, and we see the ESBL showing up between as an interaction, as a synergy between clavulanic acid and the third generation cephalosporinases. Why is this important? Well, because we do not have cloxacillin in Romania. Unfortunately, it's not available to us as a molecule. It costs very much to import. It is not available in pharmacies, and then you would end up with very, very expensive media, even though you would ultimately like your media to actually screen for something that is, and screening is supposed to be something that is quite cheap. So one of the things that we did as an internal project in the Cantacuzino Institute is that we characterized the use of oxacillin. And oxacillin is quite readily available. We have a national producer for oxacillin, and we titrated the level of oxacillin to match the activity of cloxacillin, and we produced a media that would be able to allow us to screen such bacteria as well. And these are some representative examples of cloxacillin, oxacillin medium being tested in between the two. But when you come to a carbapenemase, of course, oxacillin is not going to be sufficient anymore, so we would be required to test even further. Is this plate that isn't telling us much actually from a carbapenemase producer, or is it something else? Are there impermeability? Are there efflux pumps at work? So this is where we come to expertise, and expertise means that you have to look at all of these images, and they all kind of look the same, but one is not a carbapenemase, and you have to know which is which. And for this, the Pasteur Institute actually uses a very nice little flowchart, and then we get to the choice of carbapenemase detection methods. And we have our small contribution here, the RSIM and the RSIM-A, the rapid carbapenem inactivation method, which is a derivative of the fundamental work done by the Dutch group and then replicated in Pasteur, which actually allows us to turn a 24-hour test in which we would grow the bacteria in the presence of a disc of antibiotic, which would or would not be inactivated by the bacteria, into something that takes a little over or around 3 hours, 2.5 hours. Why is that important? And why am I presenting this to you here? Because it's all, again, done in microbial dynamics. In fact, what we've done is that we're no longer looking for the growth of the indicator strain of Escherichia coli, which is used with the antibiotic disc that we're adding over the bacteria. We are actually using this supernatant, and we're adding it over a live bacterial suspension in a broth of E. coli. And we're just using a simple laboratory nephelometer to see if the bacteria continues to grow or not. Basically what we've done in microcalorimetry, there we had responses at 1 hour. Now we're using the nephelometer for at least 2 hours, but it gives us a very, very good information. And we've studied this process, and we've basically published the RSIM protocol. The RSIM-A has been published last year. We got a very nice response from one of the developers of the whole field of carbapenemases, and we're working with some of the best people in carbapenemases, and now we're trying to have a step further. These are results that we've presented at ECCMID. We are constantly tweaking, we are adding inhibitors, we are adding cofactors, we are incubating with antibiotic at different times. Right now we're using a lysis buffer, which we've not used up until now, to get the bacteria interacting with the antibiotic as quickly as possible, so that we get to that desired near real-time antimicrobial antibiotic susceptibility and resistance mechanism detection. So this is RSIM imipenem. and on a very, very nicely characterized database of Enterobacteriaceae. We have very good results, but you're only as good as what you test, and you can only develop when you challenge your method. And so, even though we've kind of made our own little flowchart and we've adapted oxacillin for in the state of cloxacillin, and we're using the RSIM instead of the carbo NP. Real life always likes to throw us a curveball, and this is absolutely normal, and this is the challenging and the fun aspect of microbiology, because you're always challenged by what nature is actually sending your way. We've just had a strain that flagged positive, and it flagged positive especially because it was resistant to a novel antibiotic inhibitor combination, so ceftazidime avibactam, not so much because it was positive in the ertapenem or the meropenem screening, which were quite difficult to spot. And even in the best of cases, when you would use an immunochromatographic rapid test, these cost $15, and they can give you the information in about 15 minutes. You see, per protocol, when you test it, there is no information. When you raise the inoculum level, still, I didn't see it. It was a colleague of mine, probably better eyes, that came in and said, look, don't you see a faint line over there? I was, no, I don't think I do. But he said, look, let me test it with a whole lot. Let me clog up the test and see if there's something flagging there. And indeed, it was. So as per Professor Kifiluk's comment on expression levels and detection, that this is something that is challenging to any laboratory. And this test, in fact, this strain, in fact, was negative in RSIM. And now we've worked on the RSIM, and we're hoping that we uniform it with for different types of bacteria, and we got this to test positive. So, using very simple methods that can be found in clinical laboratories, just discs of antibiotic, a little bit of broth, the E. coli ATCC 25922, the standard ATCC drug susceptible. We can check in less than 3 hours the presence of carbapenemases, and we can act. And what do I mean by acting? Well, this is what history teaches us about levels of carbapenemase production in different countries. It is difficult to judge where we are today in Romania, and I am very proud to have worked with Professor Kifiluk and her team on a protocol that actually is showing us the pollution, the real life dissemination of these enzymes in Romania. The paradigm that we are trying to change is that enzymes act very fast. Bacteria grow slower than the activity of enzymes. Very expensive equipment is not readily available, certainly not in hospitals, maybe in research institutes. We are trying to bridge the gap between the enzyme field and the bacterial field, so that we can actually show the very, very fast kinetics of enzymes, even the ones that we would call slow hydrolyzers, in the least amount of time possible, using the cheapest methods possible. Because of course, we can do this with qPCRs, we can do this with different types of expensive equipment. The contribution that we are here to tell you about today is that we are actually doing it with very basic laboratory equipment. And this started as an extremely simple test, just some water and an antibiotic test. But it has evolved. It has evolved into something that it may not be perfectly compatible with absolutely every laboratory out there. Because you need some zinc, and you need some oxacillin or cloxacillin, but we have oxacillin in Romania, and zinc is quite cheap to buy. And it's interesting how we managed to adapt. And as I said, we have to take action. We have to report. we have to optimize our antibiotic usage. In the hospitals, we have to implicate more in increased hygiene methods and dedicated personnel, and we have to do tracing. And we hope to be able to do this in the near future. And I am extremely pleased to have talked to you in front of you today, and if anybody would want to contact us further on, there are emails which can be always accessible.
Thank you very much. Question?
I mean, a general comment and question, because you discussed about action, and I think you're also in the medical system. I lived in the past 10 years in the UK, and in the UK, and this is also because, actually because of we are on the red map. And in the UK, I've got children, right? And I've been a child in Romania a long time, not so long time ago. So in the UK, you're not getting antibiotics for 3 days if you have fever. So only after 3 days they consider you giving you antibiotics. I know in Romania we were given antibiotics immediately after before. Even before. Is there anything like this? I mean, do you think this is an effective measure to counteract this redness on the map? Question number one, and question number two: what actions are you taking if this is an effective measure? Or what other actions are you taking to counteract this redness on the map? Obviously, this is an action, but I think this is not so. This is treating, not attacking the root of the problem.
So if I understood firstly, the first question was about would the RSIM and the development of such techniques help us in reducing antimicrobial consumption in hospitals and in
my question was, I mean, guiding the doctors to delay antibiotic administration. For example, after 3 days of high temperature, would this be an effective measure to reduce antibiotic resistance in Romania?
This is something that we in the medical field would call antibiotic stewardship. So there are some people to whom, based on risk factors, a fever is much more severe or has the potential to be more severe than others. So neutropenic patients, very young children, the very old. These are people in which a fever would be treated in the UK and in Romania quite more hands-on. Okay, so at least they would be in a hospital or in a intermediate facility, and they would be monitored much more intensively for the need of antibiotics. For the general population, of course, there is an extreme need to talk to pharmacists, talk to prescribing doctors, to physicians, to general practitioners, and have these guidelines in place that you have mentioned. A well-off person beforehand with a fever should not get antibiotics for 3 days. That is conventional and that is okay. For somebody that is frail, not getting.
I was not referring to someone frail.
Yeah.
I was referring to.
So these guidelines exist.
So they exist. They exist today. In
yes. They are, part of what we teach students. They are what people say, and of course, face to face at 3 o'clock in the morning, and somebody's coming into the ED telling you that they have fever. It's a very different impact that you got. But I understand, and yes, actually applying the guidelines is a very good place to start. Your second question pertains
I think this answers my question. The guidelines exist and they are clearly,
So there is nobody imposing that you should not give three days of antibiotics. But this is antibiotic stewardship. We are talking about antibiotics. We know about antibiotic stewardship. It's difficult to implement because some people in Romania, fortunately, still have that idea that the doctor didn't do anything because I didn't give him anything. I'm going to stop it right here because it's on the internet.
But that is the public awareness you can think is. I mean, this is public health.
That's what I'm saying. So this is public health. This is
not the doctors necessarily, but as you say, patient comes to you and say, "What do I do? I have to give him something," or so they think I'm not a good doctor. This is really psychological. So
this comes from our history as well, since most of the times, if somebody, God forbid, died, the moniker that the doctor was using, well, I did everything I could. I gave him antibiotics, I did everything I could, I tried to save him, nothing worked. So it's very difficult to handle these drugs which are available to specialists and non-specialists alike. Specialists, I'm talking about infective disease doctors. But everybody can wield antibiotics. The GP, the pharmacist, everybody can wield antibiotics. And so, yes, they are a common good that we need to understand and respect, and of course, try to work with it for as long as we can. Here, we are talking about a very particular class of enzymes and divergence of mechanisms that lead us to much more frequent therapeutic endpoints. It's one thing to give penicillin, it's something else to give carbapenem. And yes, hospitals are applying this, and I know of very many hospitals in which the policy is that yes, you are allowed as a physician to treat the patient that you want with a carbapenem for 3 days, because somebody in the hospital is already considered more frail or has something that flagged for the clinician. But afterwards, to continue the carbapenem treatment, you have to get the signature of an ID physician, and this is perfectly. So it's outside the hospital, inside the hospital, inside the environment.
One extra thing, and in relation, I think that the institutes should maybe do more public outreach. The institutes like yours, right, to educate the public that actually antibiotics are not something. But anyway,
The new
for
building,
discussion.
the new building for hospital are necessary in this case?
I think it is very, very difficult to eradicate these resistances which have built up in the old buildings, and the circuits and the fluxes of patients are not amenable to what we know today. So yes, it would be great to have new hospitals. Yes, please.
Yeah, Professor.
Professor.
So I was, I don't know, tamed by the idea of having everything simple. So my difficulty in understanding the simpleness and the time frame of your essay is connected with using 1860 cell lines, because that for me is incompatible with analyzing samples from patients, so the ones who are actually revealing the resistance to certain antibiotic. Then I have slight difficulties in understanding the concentration of bacteria you are using, and that is connected with difficult to cultivate bacteria with mixed cultures. And I also have difficulties in understanding the dynamics you showed about temperature variations, which were very far from an ideal sinusoidal or sigmoidal distribution.
Yes.
So one question, how do you handle mixed infections? How do you handle the concentration needed and the specificity of patient?
Thank you very much. It is true that we have not applied this method as of yet to patient samples. We are working on what is one of the most important patient samples and the one that is less frequently mixed. That is to say, we are applying this to blood cultures, and we are trying to figure out the optimal parameters in which a blood culture which would flag positive at 6 hours time would be tested through these methods and give you antibiotic resistance detection and another 3 or 4 hours time. This is something extremely difficult to do, and the UCAST has actually proposed very simple guidelines in the RAST methodology. So I'm going to do for the carbapenem screening. They take a culture bottle of blood which has flagged positive in a machine, and the machine reads it as quickly as something is determined. And they said, if it's flagged in the last 4 hours, just drop 150 microliters of the blood onto the agar plate, and you read it in 4, 6, 8, and 20 hours time. So the UCAS did this on many thousands of bacterial species because they are the European Union Commission on Antimicrobial Susceptibility Testing. They do the guidelines. And they actually made this for our help. In mixed cultures, it would be difficult because you need an inoculum to begin with, the amount of bacteria that you test for in the beginning. And we know from literature and testing these bacteria on our own that we can go to as low as 1 microliter loopful. A loopful of 1 microliter of bacteria culture to be tested and screen positive for a carbapenemase, detect actually positive for a carbapenemase is amazing. Because you have more than you would need for a 0.5 McFarland index, and that's 10 to the power of 8 microns. So 1 colony is enough. As to the variations in temperature for the sinusoid, these are going to the signal here. Perfect. This is actually because in the calorimeter we have 2 different metabolic processes of the bacteria happening. So we get 2 humps because we actually have 2 populations of bacteria. We actually have a bacteria that's growing in the dissolved oxygen in the medium, and the bacteria, let me show you in this first one because this was for full growth of the bacteria. This is growth of bacteria with dissolved oxygen already inside the media, and then the media gets into a lack of oxygen, and only at the interface where they have enough air from the amount of enough oxygen from the amount of air on the upper side do these grow. So oftentimes microbiologists would come in and they would see a veil of bacteria growing at the top of the bacteria of the liquid culture, because bacteria, some of them, love oxygen.
No shaking indoors
here. Here, no shaking at all. No shaking. So only passive diffusion of the oxygen into the medium. And this hump, we've proven this using metabolites that affect bacteria growing in anaerobic conditions, so metronidazole. We've tested this with paraffin. We just sealed off the top half of the ampule, and you would only get this part of the growth when you have no more air diffusing, and you would get only this part of the growth when you have metronidazole, and you would basically inhibit some of the bacteria that are growing anaerobically. As opposed to your third question, I think, about the ATCC line, this is used only as an indicator. So I'm testing my bacteria. I'm testing my bacteria here. It's metabolizing the disk of antibody here. And then I'm challenging an indicator, which is the ATCC E. coli bacteria, which grows extremely fast, at a McFarland index of 1, and we are seeing it grow in 2 hours. So that's very simple to do.
But you are assuming that you have enough enzymes coming from the supernatant of your initial solution.
Do OXA23 say anything to anybody here? OXA23 carbapenemase? I'm sorry, Professor. This is a very slow lytic enzyme. This does not produce carbapenem resistance. This causes resistance to amoxicillin in Acinetobacter, which is also very impermeable and has a very, very important MC. This does not flag in the conventional tests for positivity. OXA23 flags positive in our test, and I'm extremely happy to say this. And we've actually had a very difficult coming, when you're small but you're on the shoulders of giants, you have much difficulty going to the people that produced the tests and said, look, I think there's something here that you didn't found before, but it constantly flags positive for me, and I think there's something in here. Somebody says, okay, send it to NGS, and then somebody else says, yeah, there's a carbon dependent in there, yeah, good catch. And these are the tests, and these are the enzymes.
I'm sorry, the discussion is very interesting. We can continue separately.
Tomorrow at the round table.
Round table.
Okay, Professor Kitty, you have a question, please.
No, I have just wanted to underline that.
Comments,
Yes,
okay.
yes, this method is applied after we have isolated the bacterial strain. So we have already lost 24 hours. And look what beautiful work, but for only one mechanism of resistance. And for only one type of strain, and for only one type of antibiotic. And we have vancomycin-resistant enterococcus, we have hypercoccus-resistant aminoglycosides, we have methicillin-resistant, and so on.
Yes.
So we need some general method, very easily tunable for different antibiotics. And
it is possible that something like microcalorimetry, which actually shows you the growth of bacteria, may do something much faster. But for carbopentinases, we know that the enzyme is the problem. It's no longer microbiology, it's chemistry.
Okay, thank you once again for very convincing talk. Thank you very much. Now I would like to invite Dr. Sabina Zudak from the University of Medicine and Pharmacy Carol Davila Colentina Hospital, University Hospital, and the Zaya Artificial Intelligence cellular.
Good evening. Thank you very much for inviting me here to speak in front of you. I'm honored and a little bit anxious. I hope in a good way. Seeing the previous presentations, my presentation differs quite a lot. In fact, I'm the representing of a large team from Colentina and Zaya company, and we try to do something which is not very often tried, and I mean we try to develop some algorithms based on artificial intelligence in order to identify things in histopathology. We all know that AI is developing methods based on AI are developing a lot in the medical field, not so fast and not so much in histopathology. Maybe we pathologists, I'm a pathologist, maybe we are a little bit retarded, not very open to the new things, but also we know that the histopathological diagnosis is the medical diagnosis with the highest degree of certainty. In fact, in many textbooks is written something which I really hate, that the only perfect diagnosis is the histopathological one. No, please, I'm not God. Only God is without mistake. I should be allowed to make mistakes. And the mistakes in histopathology are very difficult to correct. If you are saying to someone that he has cancer, it's difficult to change the things later on. We try to do something which in the beginning I thought is simple, to identify acid-fast bacilli, meaning Mycobacterium tuberculosis, on Ziel-Nielsen stain slides. Later on, my colleagues, the machine learning engineers, they hated me for this idea. I thought it's so easy to look for a red rod, a red stick on a blue background, but in the end it seems that it was not so easy, and I will show you why. Why we choose tuberculosis? Well, tuberculosis is very old. I was astonished to discover that there are hundreds of millions of years ago evidence for mycobacteria. Also in humans, up to 3 million years ago, they are arguments that they had tuberculosis. In the moment, we have almost 10 million people with tuberculosis worldwide. It is the first cause of death by infectious disease worldwide. In the well-developed countries, there's not so much tuberculosis, but neither West Europe or the United States should think that, okay, they are spared. There is a lot of migration these days, and also the COVID impact is disastrous. In fact, in India, half of the tuberculosis, the patients with tuberculosis are not diagnosed to date, just because of the impossibility to get medical attention. Okay, this was a case from an infant, a boy 1 year old, nodule on the thigh, granulomatous paniculitis, extremely easy to identify, millions of bacilli. Everything which is red here is Mycobacterium tuberculosis. Okay, what about this? This was a lady, 60 years old, a small piece of skin, 12 square millimeters. We examined 4 sections. She had granulomatous inflammation. In 4 sections of tissue, these were the only 2 bacilli that we can find on a slide. So something like this is easy to miss, so we have a problem. What could be the solution? More sections, more slides. This is awful. It's more time to examine the slides. And we know that doctors are fewer every year, and pathology is a not very appealing specialty. One of my interesting cases is either a dead person, or a very rare tumor. The patient will die in about 6 months. In fact, he's already dead, but I'm forced to tell him or her that he's dead. So it's awful, and very few people want to become pathologists. Everyone wants to be cardiologists, gynecologists, or neurosurgeons. So a solution could be artificial intelligence. It would increase the number of the slides a pathologist can examine. It will save time and money required for different investigation that the patient is not doing if he or she has the diagnosis. And also, very important, it diminishing the interval between the moment of the biopsy and the beginning of the treatment, because we want a very fast diagnosis in order to start as soon as possible the treatment. What have we done? We look in the archives of Colentina Hospital for about 12 years. We selected more than 2,000 cases with Ziehl-Neelsen stains mentioned in histopathological report. All these cases were evaluated. We looked both on H&E and Ziehl-Neelsen stain. We selected case of positive cases. They were cases reported positive in the histopathological report and reconfirmed as such on the microscopic evaluation. And also we have the negative cases, cases without acid-fast bacilli in Ziehl-Neelsen stain, and confirmed diagnosis of other illnesses than tuberculosis. Why? Of course, we excluded consultation cases because the paraffin block was returned to the patient and we don't have material to perform new slides. If we have discordances between the initial histopathological report and evaluation, we just dropped that case. And also, we excluded cases with a histopathological appearance highly suggestive of tuberculosis. Why? Every medical student knows that tuberculosis is a specific inflammation. Otherwise, if I'm looking to a slide with tuberculosis, based on the histopathological appearance alone, I should say that that patient has tuberculosis. The lesions are specific. It has granulomatous inflammation with giant cells and epithelioids and caseating necrosis. However, real life is not as simple. There are a lot of lesions similar with tuberculosis. They are not tuberculosis. We should perform several special stains. In some cases, we should perform immunohistochemistry, PCR, and sometimes we need a very good correlation with a lot of medical tests, radiographic images, quantiferon whatsoever. And when we found cases highly suggestive of tuberculosis but with negative Nielsen stain, they could be paucibacillary lesions. If I have only 1 or 2 bacilli in a piece of tissue, something like this, 2 per 3 centimeters, 6 square centimeters, it's quite difficult to examine and to look for a bacilli who's 3 or 4 microns in length. Also, the bacillum is very thin, only 1 micron, and the section has 3 microns. So if I'm cutting a new section, maybe I can find another bacillum. Okay, we were very specific in constructing our data set because the rule in pathology is garbage in, garbage out. So if you are giving to something, no matter what, some things that are not good, the result for sure will be not good. We scanned the slides as whole slide image to have the complete section on the slide. The format was SBS. We used 2 scanners, a manual scanner provided by Microvisioner, German firm, and an automatic scanner from Leica. And we selected the group. It was one training group with 510 cases, 25% positive, 400 cases, 3/3 negative. And this was the 60 case group for clinical testing. 23 cases positive, 37 negative. The VSIs were annotated by 7 pathologists with 4 to 33 years of experience. I'm the person with 23 years. I'm the older person from our group. And we identified positive areas. We begin with the in-house annotation platform. Later on, we switched to Cyphomin platform, and I will show you some images. We selected bounding boxes or point line annotation, and all the positive areas were extended to a 64 pixels bounding boxes that completely included the positive markings. We ended in having 263,000 positive bounding boxes. In fact, today is the largest data set for mycobacterium existing in the world. The negative samples were easily to obtain from VSI's negative cases, but this was easy. You can get as many negative bounding boxes as you want. Also, we annotated manually negative areas inside the positive cases in order to have a larger variety of colors. Cellular stain is a manual stain. It's not possible to perform it in an automatic machine. You have to boil the dye on the slide. In fact, to explain you easier, in difficult cases, I'm asking for 2 Nielsen slides performed in 2 different days. So in the same laboratory, with the same technique, the same technicians who's performing the stain, I'm asking, okay, do it today and do it tomorrow. And every time we obtain different results in color. in a lesion with a lot of bacilli, it's not a problem. But if you have only 1, 2, or 3 bacilli and you are looking for the needle in the haystack, then it's better to have as many possibilities as humanly possible. Okay, this was the in-house platform. We made squares, small squares, some of them with 1 bacilla inside, 2 or many. This was cytomine, much easier to use, just putting dots here on every red bacilli. These were the negative areas. Look, this is another case. Please appreciate the great variation in the color. This is a negative case, just the red blood cells are a little bit pink. This is an internal control. If you don't have pink red blood cells, then the stain is not good. Okay, we later on the machine learning engineer perform several augmentation techniques exactly in order to enlarge the diversity of the staining process. You never know how it will look a stain performed in another laboratory. We also change the position in the bacillum in the bounding box. From what I learned, the neural network is tricky. If you are providing it just bacilli in the center of the bounding box, she will learn that, okay, the bacilli should be looked for only in the center and not in the periphery. So we shifted the position from here to here in order to obtain different position of the bacilli in the bounding box. Also, a lot of alteration in the brightness, contrast, saturation, whatsoever. In the end, we begin with almost 1/4 million positive bounding boxes, and we ended with 2.5 million. Okay, these, I'm sorry for me, are just words. We use a deep convolutional neural network. The name of it is this one. I understand it has the state-of-the-art performance to date. And well, it did what it should do. We obtained different models, and we selected, okay, this is the training part. We started with various proportions of positive and negative in the data training set. We limit our procedure to 100 million samples. I understand that the neural network is bored at one moment and doesn't want to look too many. It's saturated. And the results for each model were validated on another validation set comprising 280,000 pages, of which about 5% are positive. The main problem in identifying bacilli in Thionine cell stain is to fight with the artifacts, with the positive things which are not bacilli. If the network is showing, okay, this is a bacilli, I'm very happy. But if it's showing me 10,000 patches with junks inside, I'm losing a lot of time, and okay, I'm saying, okay, Zaya, you are a very good girl, go away, I will look to the slide myself, and let's finish the job. Okay, these were the results in the beginning, a lot of patches with a lot of numbers on them. The numbers are probabilities, but these are not probabilities, these are probably positive patches, these are probably negative patches. They are very, very large, it's a digital enlargement, so this is why it's not very clear, but here and here you can see they are bacilli. This probability is not the probability to have bacilli, but to belong, to be similar to the positive data set we feed it the model in the training moment. The results were better and better. These are the results, this is the area under the curve, the aggregated area under the curve 1 month ago. It's about 0.968. 2 months ago we had 0.89 and I was very happy. Then we reached 0.92, 94, and now it's almost, it's 0.96. How it's working? The pathologist is scanning the Nielsen slide and it's uploading the BSI in the platform. The algorithm is giving the results and the pathologist analyzes the results and decide the diagnosis. So it's just a tool to help the pathologist, and it's not making diagnosis instead of pathologist. However, this is a case with very few bacilli. In this area, Zaya showed us 2 bacilli, this and that. Positive score 0.94. It was a slide, a paucibacillar lesion, and this was the second patch showed by the system. So the diagnosis was okay. In the first one, nothing. It was a mistake. The second one, we have bacilli. The slide is positive. 10 seconds diagnosis. However, we had a lot of jumps. Look here. Positive score 0.96. Wonderful. Maybe here. Let's get rid of the green square. In fact, it was a red blood cell. Only part of it was in the square, and the algorithm identified just what's in the square, and it discovered a red line, short, and said, okay, it's a bacillus. No, it's not. Another problem was like here. Look, without the green square, this is a mast cell. The methylene blue from the Nielsen stain is staining in purple the granulations from the mast cell. This is a mast cell, and this area here was inside the patch, mimic a bacillus. We take all these patches and we said, we label them negative or positive, negative or positive. The informations were re-entered in the model for training, and we obtained an aggregate area under the curve of 0.977. I was incredibly happy, and I really thought, okay, this is too good to be true. So we went further to a testing process, man versus machine. In pathology, most of the pathologists are women. We assembled 4 teams of pathologists with similar, 2 pathologists with similar experience in each team, a senior pathologist with more than 10 years experience, younger senior pathologists, specialists, and residents. We asked the pathologists to look to the slides to the microscope, at the microscope in the usual way, to look to the BSIs, to the scans, and here we have a big problem. There are studies in the literature saying that there is a discrepancy between the diagnosis made by pathologists on scans versus diagnosis made on slides up to 63%, which is huge. It's almost like using a coin. Of course, the algorithm-aided diagnosis. We had a washout period of 2 weeks. We intended not to cheat in any way. And of course, the golden standard was the original histopathological report reconfirmed by H&E and Thiel-Nielsen. I was not part of the teams who were involved in the testing process because I was the person who coined the testing group, and my colleagues hated for this. Why? Well, these are some data. This is why they hated me. I did not choose cases easy to appreciate on Thiel-Nielsen saying that, okay, this is not TB. It's not possible to be TB. It's just a reactive lymph node, or it's just a squamous cell carcinoma, or whatsoever. No, I looked for necrotizing granulomatous inflammation, cat scratch disease, some unspecific granulomatous inflammation, granulomas in sarcoidosis. You can have tuberculosis with a lot of granulomas in differential diagnosis with sarcoidosis. Toxoplasmosis. Toxoplasmosis is filling your lymph node with epithelioid histiocytes, and epithelioids are marking of tuberculosis. A lot of necrosis, Kikuchi disease, necrosis in lymphomas, in carcinomas, necrosis, coagulative necrosis, also florid histiocytosis. I did not want that the human mind cheat just to look to the slide and say, okay, this is negative because I was not able to see bacilli, not because I'm sure this is not tuberculosis. Okay, these are the results. The first results, let's look to the accuracy. We have here, these are the experienced pathologists, senior pathologists, younger senior pathologists, specialists, and residents. And this is Zaya. Well, honestly, I expected the best results to be somewhere here, to the specialists. A lot of experience and also a lot of patience. For the senior, very senior pathologists, I expected lower results. To my surprise, not really. It was quite in the same spectrum. The residents had poor results. I thought, okay, let's look how experienced were they. No correlation. It was, however, a relation with the experience of exposure to UVSIs. The first 6 pathologists, without the residents, they were involved in the annotation process. And first of them annotated for about 1.5 years. And they were used, they had the training curve to learn how to look to VSIs. Okay, it's what I said, that we have a statistically significant association for specificity and accuracy, not for sensitivity. Really, I don't care too much about sensitivity. I want a very good, a very high specificity, because I want that the algorithm, when it's saying that this is tuberculosis, to be tuberculosis. In pathology, in medicine in general, but in pathology, you don't want to overstate. If I'm telling to a patient that he or she has tuberculosis, it will follow 6 months of very hard treatment with antibiotics. 2 times a week, he'll receive something like this, a full feast of drugs. The patient will take the drugs for the first month. Later on, he will feel better. Maybe if he's very compliant, he will take another month, and then he will discard the treatment. And now it's the way we ended with so many multi-drug resistant cases of tuberculosis in Romania. Okay, what about performance on slides? The accuracy of the pathologist was much better. In fact, there was one senior pathologist with the same accuracy, the same, in fact, both Zaya and this pathologist made only one mistake. they did not see a case which was positive. All the negative cases were labeled correctly as negative. But the most important advantage was when we are looking to the time. How much time the pathologists consume looking to the slides. This is the time used for BSIs. You can see there were pathologists looking 80 minutes for a slide. In here, 50 minutes. I have to tell you that last year I received a consultation, a case with 7 arcing blocks, huge pieces of tissue, 3 per 2.5 centimeters of tissue. The differential diagnosis was between Crohn's disease, inflammatory colitis, and tuberculosis. I performed Ziel-Nielsen on each slide. For those 7 slides, I spent 2 days looking at them, and I ended in discovering in one Langerhans giant cell 2 bacilli. One giant multinucleated giant cells has around 100 microns, and I looked in 6 or 7 square centimeters of tissue in 7 slides. It's obvious also that our pathologists are trained to examine Ziel-Nielsen stains, which is also important. If you take a normal pathologist, not so used to, I will finish in 3 minutes, sorry. If we look, a normal pathologist is not used to and will spend more and more time to look to the slides. What did Zaya? The average time was less than 2 minutes. In fact, it's not what did Zaya. How much time the pathologist used by evaluating Zaya's results and decided the diagnosis. We looked to the errors. I was astonished. 31 cases had errors from 60, and it was 1 case, a positive one, in which 6 pathologists said that is negative, and in fact it was positive. This is a case with 5 false positive. Here, as you see, it seems that maybe it's something. This was cat-scratch disease. I know that it was cat-scratch disease based on immunological markers, and also this is not caseatic necrosis, it's furation. This was missed as it was false negative. Half of the pathologists said it's negative. Here you can see clearly there are few bacilli, but look how big is the tissue. This is the case, very difficult, 6 negatives of 8 examination, on the only false negative of Zaya. Here we have 2 bacilli, but very faint, very difficult. I have to admit that when I looked again at this slide, in the beginning I saw the bacilli, there are other levels with bacilli, but when re-examining the slide, I was not able to find them in the first 15 minutes. Then I began to look the next day in the morning, very happy and with a lot of coffee inside, and I was able to find the bacilli and make a picture. This is a short, well, we look to the literature. There are only 4 papers with attempts to develop algorithms based on IE for detecting tuberculosis on tissue. The results are different. This is a very good study. These 2 studies are good, but they have, this one has only 600 positive bounding boxes, 500 positive. We have almost 250,000. Pantanovic has more cases. His results are very good. The sensitivity is not very good. A lot of positive cases are missed. but the specificity is good, this is awful. If I'm getting a specificity of 83%, I will end in say, okay, I don't like the algorithm, I prefer to do it by myself. And few examples, positive score 0.89. Of course, this is a positive here, obvious positive. Here, it's not obvious that there are bacilli. If we are looking, okay, this is. It can show you many patches, but this is where the algorithm is very important. Many red blood cells, and here a patch with bacilli. What we do now, we started to develop a model for diagnosing epithelial carcinoma. I thought, okay, if the machine learning engineers were unhappy with the small, tiny red rod, what to say about carcinoma, they are happy. It seems it's much easier to identify epithelial carcinoma than mycobacteria. And this is our team from Valentina and Zara. And thank you for your attention.
Next question, comment.
Yes.
Oh, well, mine was really a comment. So congratulations on the audacity to bring CIA, so criminal investigation into action. I was wondering, you were mentioning so many bacteria in one case, and on the other ones, very few of them. Do you feel that the degree of tissue change in relation to the actual infection? Because I thought, how come that the bacteria are inside the tissue and the tissue itself is not encapsulating, not showing signs of immune surveillance and so on? And whether this type of information is somehow taken into account by the automatic procedure to analyze the image to increase the accuracy of the investigation. So,
Okay, there are multiple questions. First, yes, there are huge differences between the tissues with tuberculosis. It depends mainly about the immunological capacity of the patient. It depends if the patient is immunosuppressed, and it depends about the specific type of immunosuppression. In tuberculosis, you have two different types of reaction. The proliferation one, a specific granulation tissue with giant cell granulomas with epithelioids, and the necrosis. The first part you are obtaining by a proliferation of activated macrophages. The second one you are obtaining by an enhancement of the cytotoxic lymphocytes, which are destroying the naive macrophages, which are harboring the Koch bacilli, and without the production of specific trained macrophages able to destroy the mycobacteria. Okay, so we have differences here. Second, I went very fast, but you can have tuberculosis in different types of tissue. The problem with the automatic diagnosis in histopathology is that you are, well, an AI algorithm for diagnosis in histopathology is not a sausage factory. In a sausage factory, you are putting different types of meat and spices, and you are obtaining different types of sausages. It's like preparing a line for developing vaccines against influenza. It depends about the virus you are starting with, and you obtain different types of vaccines. It's more like the vaccines against COVID with different methods of vaccination. If I want to cook soup, I cannot use and the soup and the cake, I cannot use the same receipt. I can take the ingredients from the cake, I put in the receipt for soup, and I will not obtain a cake, neither a soup. And so you have to have a specific algorithm for each disease. And in fact, in order to diagnose multivariate carcinoma, you have to assemble, to our estimation today, 13 algorithms, different algorithms. So if we were trying to design algorithms to make diagnosis of tuberculosis, it was something different. We went only for identifying mycobacteria, the bacilli. Okay, so this is one.
Okay.
Okay, but now I have a follow-up. So the
Yes.
training set was the same type of tissue, or was it
No,
any kind of
no,
tissue?
no,
And then
no,
you
no,
were
no.
looking at the granulomas, which all look
We
more or
had
less the
had
same?
tissues from skin, from lung, from lymph nodes, from soft tissue, from GI tract. So yes.
I have a question. You mentioned there are a number of algorithms.
Yes.
Does it matter in which sequence you apply them?
Sorry.
There are a number of algorithms.
Yes.
Does it matter in which sequence you apply them?
No. We had different algorithms, and we choose the best one.
No, I was meaning the one algorithm has different procedures, filtering, thresholding, and so forth.
If we want to do a diagnosis for biodynamic carcinoma, we should apply all the algorithms.
Yes, but one algorithm has several steps.
Yes.
Thresholding, enhancing, support, and so forth. Does it matter in which order you apply these steps in a receipt?
We are not using the results from the first algorithm to be fed in the second one.
Let me ask you the other way.
Professor
So,
Melinde.
I understand the question.
Yes,
question,
please.
I understand the
Actually,
question is
I'm
how
a
you
pathologist, so.
Yeah, so to translate, and
Yes.
maybe I'm not, because I didn't understood the question I see. So how you benchmark basically when you have to test different algorithms, how will you benchmark them, or how you optimize with respect to what? To the area under the curve, F1 score, how you choose the best, what is the criterion and choose the best?
In order to choose the best model for one algorithm, we look for the area under the curve.
This is for
Okay,
me,
this is for one algorithm with several models. But if we have several algorithms for one disease, those algorithms will be applied on the original PSI. And we will obtain different types of results because we have to look for, invasion, nuclear pleomorphism, mitosis, I don't know.
But this is why it's very common at the beginning when you present results, numbers looking great for that. I go closer and I look at many scores on your confusion matrix, it was fine. But when you compare with the others, you have to tell us, what they've been using, how they treat the.
Well, I think it's a matter
in the end, there are numbers, but these numbers are obtained in a very different way. So you have to be very careful because, 90%, 92% it depends how it was obtained, and it's very tricky. And
it's true, and we are quite confident. In fact, now I'm more confident after the clinical test. But we were quite confident because the other authors had some of them, they had very few positive cases. One of them had only 6 cases
So
positive.
you are saying they were treating, databases with
The
huge class
data,
imbalance,
yes, the data set was too small, and
but small and with a lot of class imbalance between positive and negative.
they were not so many. 600 positive and almost 1,000 negative. This is very small.
Yes,
But you have to have a very good and patient pathologists to annotate them.
This is for sure, but I think it's very challenging because you have to benchmark and there are methods how to test really the algorithms you are using.
Honestly, this is why I'm not saying that we should take Zaya's results as granted. They should be verified by pathologists.
Okay, thank you.
Sorry.
You can continue after.
Yes, thank you.
Yeah, thank you once again. No, round table, please. Eugen.
I think we should allow ourselves 5 minutes and